Okay, I think we'll get started. We'll see if anyone else ends up trickling in, but I guess since we're kind of off schedule a little bit, we might not have as many people show up today. Before we get going, does anyone have any remaining questions from the last class? I know there's quite a few people that were having issues getting the SRA toolkit to download and work.
I have a question, but maybe we could like save it till the end. When I was downloading the SRA toolkit, I was just like copying pasting from the slide. So I think I use your UID rather than my URD when like appending to the bash RC folder. So I was wondering if there was a way I could get like rid of your UID or like delete the last line of the bash RC. Like I tried using the cat command on it.
Okay.
Yeah.
Yeah, so we can edit that file with Vim.
Okay.
So if you're on your home directory and just type vim.bash rc, and I guess I don't have mine here, it's in a different file, but if you scroll down to where that line was added at the bottom and
Okay.
enter insert mode, you can modify that path so that it has your UID instead of mine.
Okay. Thanks.
Yeah. Are there any other questions from last time with where people got stuck or wants to review?
Thanks for replying on Slack, Jordan. I'll give that a try and see if it solves my problems.
Okay, sounds good. And you're running in the bash shell?
Yes.
Okay.
I think.
Okay. Maybe if that doesn't work, check your shell and just make sure it is bash. I don't know if that.
Oh yes, sorry. This was the thing where I had to go to CHPC and change my. Was that? That's what you mean by bash shell?
Yeah. If you just yeah.
Yes, I definitely did that, and so I think I am in bash.
Okay. Cool.
Can I ask you a quick question, Jordan?
Yeah.
So I had an issue with the FASTQ dump. I got an error saying FASTQ dump 2.10.7 error, name not found while resolving query with virtual file system module.
Interesting. Okay. I
have no idea what that means.
Do you have, did you have a NCBI folder in your home directory created?
I had to create one. It wasn't automatically put in. And I tried that and that didn't solve the problem.
Okay. And I wonder if the SRA file, maybe if the output path, if it couldn't find a spot, excuse me, if it couldn't find the right spot or if it dumped it somewhere else, maybe it can't find that SRA file now.
Okay.
Let's see, so there's accession. You may try explicitly specifying the output directory with this argument.
Okay.
And then just check to see if when you run that prefetch command, if that SRA file is where you expect it to be. And then give it that same directory when you run the FASTQ dump. If that still doesn't work, let's talk some more and try to debug that.
Okay, sounds good.
Huh. Okay. Now, if there's no other questions for now, we can jump into the lesson for today. Hopefully people are starting to feel a little bit comfortable with writing a script and executing that. We're going to focus mostly on that today and specifically adapting these bash scripts to be run on the supercomputer using not just the interactive supercomputer mode like we've been doing here, where it executes right in the command line, but actually submitting a job which is run on one of the high performance clusters. And so if anyone's feeling a little bit unsure about writing a script, let's maybe talk about that quickly before we jump into this and get too far in the weeds. To just stop me and jump in if you want to talk about that. Yeah.
Jordan, I guess in my previous life, which has been mostly with a book called Python for Dummies, I've been using like text editors that are not in the terminal.
Yeah.
And so what's the deal? Like, do you think it's just simpler to do it this way, or how do you feel about that?
So usually I will write my scripts in a text editor. The reason why I'm introducing you to them is, I guess, twofold. If you just want to adapt a pre-existing script that's already on the supercomputer, sometimes it can be faster just to edit it there. And two, we're actually going to talk about this today at the end of class, is transferring files between your computer and the supercomputer. And so you'll be able to write one of these scripts in a text editor and load it into your home directory on the supercomputer. But I guess one reason was also, I guess the third reason was, since we have some people in Windows, it may be an extra couple steps transferring between, like, navigating through the file system in Windows to the supercomputer. But I think Ian, he has I think he figured out how to do this on Windows. Since I'm working on Mac, I may not know the best way to do that. So those were some of the reasons for doing that.
That makes sense.
But yeah, there's no problem writing this in a text editor and then transferring it. But since you do have to transfer it between the two systems, sometimes it's faster to debug and rerun just using Vim.
Okay. I had, I guess, two other really simple questions. I can't copy and paste like you can in PuTTY, and I also don't have like the option for autocomplete like you seem to, and it makes me really jealous.
Okay. So you can't just use this copy commands within the supercomputer?
I guess I was trying to copy like from my notes or from like, you know what I mean? Like trying to move things, or like a URL from the genome, 1,000 genomes. I had to like type the dang thing in because I couldn't copy from outside to the supercomputer and paste inside. Is that just the way life is, do you think?
That may be a security feature. I don't know. Ian, if you have any insight on this, you can jump in.
So when I was trying it, once I got to the Windows command line, like all of the commands worked for me the way that they're working for you. So like the SSH in to Kingspeak worked for me, and like the SCP to transfer, all of those commands worked. So I'm still a little unclear on where the need for PuTTY is if all of that's working from Windows.
Okay, so you're just opening the.
Okay, so I could.
Yeah, I ran the like CMD from Windows and then run as administrator the command line, and then just try the SSH from there to get into the Linux interface on the supercomputer.
Okay, and then everything works as.
And then yeah, I think
expected
once
after
you're in there, everything works the same. Yeah.
Okay. Sounds good. Yeah, that's just comes to my ignorance when it comes to Windows. So.
Ian, could you maybe share the, like I can get to my terminal in Windows, but then I guess, could you maybe share what commands I should use to make the connection to the supercomputer from there?
Yeah, it's that same SSH line for, was it Notchpeak? Is that the one that you had in the slide? Let's see if I have it.
Do you want to, maybe Ian, you can just share your screen and give us a quick walkthrough on that real quick?
I could, but I'm actually doing this on a Mac, so I don't know if it'd be useful.
Okay. Okay, maybe we'll record just a quick walkthrough on doing that later, and then we'll post that.
I'll look for it too and just paste it in the chat if I find
Cool,
it.
thanks.
Okay. And then did you have another question, or is that?
Just the autocomplete, like you, that looks like such a nice trick, but maybe I don't have that option.
Ian, are you able to autocomplete when you're in the CMD thing?
I don't know, to be honest. I didn't try that feature, but I can try it like
Uh-huh.
today, and I'll know for the afternoon session if it works on Windows. But I think so. Like I think once you're in the Linux, you're in the Linux, so everything should work
Uh-huh.
the same way, I believe.
And the way you do that is just type the first letter of, like how, it always just magically appears when you're typing, but I don't really know how you're actually doing that.
Yeah, so I can, like when you're here, so say I want to autocomplete from one of these files, I can do like cat, usually I'll type the first couple letters and then just hit tab and it should autocomplete. And this is different system to system. Like here, it'll just give me the first match and then I can just keep on hitting tab and it'll cycle through the options. Sometimes it doesn't cycle through. I think when I'm just in my Mac terminal, it'll stop at like the 1 and then list all the options. And then I have to type in the next letter or two to get to the next autocomplete.
Okay, I bet this will work for me. I did not know about the tab. I would just hit one letter and then wait patiently.
Okay. Yeah, so
I'm gonna try that.
okay. Yeah, so tab is how you autocomplete, and then like you can use the up and down arrow to cycle through old commands too.
Yeah, that one I got. Okay, cool. Thank you. That will save me a lot of typing and stupid typos.
Okay, cool. Okay, well, let's jump into today's lesson. So today we're going to talk about writing a script that you can run on the supercomputer, like one of the performance nodes itself. And the reason we want to do this is sequencing, alignment, and processing is very computationally intensive, especially when you get to running like alignment to the human genome. So because of the size, that's weird. Because of the size of the human genome, the genome index itself is usually about 50 gigabytes. And so loading that into memory and processing it is just not feasible on your own computer without your computer just getting bogged down or crashing. So that's one reason. And also it's just nice to be able to step away and let the computer do the job where you can log out, let it run, come back to it, and work with the data afterwards. So we'll write a script today that builds that genome index. I will be just sticking with yeast today, but you'll be able to adapt this to any model organism that you can find on Ensemble. And then we will write another script to actually process the FASTQ files. And once you have those FASTQ files downloaded for whatever data set you're interested, you'll be able to adapt this script to those files that you want to look at. And then finally, we will transfer files between the CHPC and your personal computer and vice versa, so that you can, maybe you want to do some kind of analysis that's a graphical interface on your computer, you'll be able to get the files that you need and use those. And then we'll just briefly go over IGV. So this is this interactive genome viewer. So you can look at read coverage and read pileups from your data compared to the reference organism. I guess depending on time, if we're kind of running short on time, we might cover this at the beginning of class on Monday, but we'll just see how things are going. And again, I'll just have this file at the beginning of each slide so that you can quickly reference where to find these slides. Okay, so SLURM. So this is the batch scheduler. So you would write a bash script that is going to build the index or do whatever, and then you submit this to SLURM. So this is what's scheduling the jobs and running them on a high performance cluster when one becomes available. And this is also helping, since we have a lot of people using this resource at the university, this also handles basically queuing your job. to run when there's space available. And this also keeps usage fair. So if you've been running a bunch of jobs recently, it may put you further in the back of the queue, whereas if you haven't run a job and the job is pretty short, it may bump you up in the queue. And so it just kind of handles balancing who should go next when a resource becomes available. And writing the SLURM script to submit to the SLURM scheduler, that is basically just a bash script like we've been writing before, but you'll see there will be a couple lines at the beginning of the script that kind of sets up this SLURM environment. Okay. And before we get into that actual building that script with those headers, one important resource in the supercomputer is called the Scratch directory. So this is a temporary file storage with, I guess, relatively unlimited space. So this is because your personal account is only limited to 50 gigabytes of data storage, but the human genome, for example, is already going to be 50 gigabytes, and per file that you're processing, you may be generating 50 to 100 gigabytes. So if you're handling one data set with 18 samples, you may be generating up to a terabyte of data. And so by sending this and kind of running the intermediate processing in the Scratch directory, this frees you from that space limitation. But since this is shared file space, it's important that you clear out these files that you don't need. So it's good practice at the end of one of your jobs to have a couple lines that transfer the files that you really want from the job back to your home directory, and then delete any intermediate files that you don't need later on. I will say one thing that I do is when I generate my genome indexes, I keep them on the Scratch directory and just store them there longer term, just because they're frequently used files. So I think there's some leeway there, but you'll once in a while get an angry email from CHPC saying, hey, people aren't clearing out the Scratch directory, remove any unneeded files. And if you don't use a file for more than a month, they will auto delete that. And I think maybe that was all I was going to say on that. But in order to navigate to the Scratch directory, so if you've never used this before, you'll have to create a new directory for your UID. So I would just, in the supercomputer, run this command. So make directory /scratch/general/luster, and then throw in my UID there. I've already created this folder though, so I can just run cd and then that, and then I can see that here is my Scratch directory with some files that I haven't, or processing jobs that I haven't cleared out yet. I have my commonly used genome indices here. And then if I run pwd, you can see that I'm now in that Scratch directory volume and not in my personal file space here. So I would, before you run any of these jobs, I would run this command with your UID so that it can actually find this file space, and it's not trying to run the job and being like, hey, I can't find the space, I'm just going to quit the job. So let's get started with building a example SLURM script. So if I were to write one of these in the supercomputer, I just do vim. Let's say we want to build yeast index. And then the suffix for this file is just .sh, like a regular bash file. So we'll open that, enter insert mode by typing I. And then we're basically going to add these kind of job setup headers at the beginning. So we will, I think someone asked a question about specifying this. This is called a shebang. I don't know why it's called a shebang, but it's this # exclamation mark followed by the location of the actual bash files that it needs to use bash. So we'll add that to the top just to make sure it knows where bash is and it's not gonna have an issue with that. And then there's a series of these #sbatch commands that we add. So any of these lines, the SLURM job interpreter is going to say, okay, this is setting up a SLURM environment variable. And this one, time, we're setting it at 72 hours. So if a job runs for longer than 72 hours, I'm just going to exit out of that command. With the CHPC nodes, as just a general user, you're limited to 72 hours for a given job to run, which is generally enough time to run a full data set of sequencing files. And if something's going longer than that, probably something hit an infinite loop where it's just stuck at a step and it can't find a way out. And so that's just the way of the SLURM manager freeing up that space for someone else, because more than likely there's some issue with the script where it's just not continuing. We will just request one node. And so in general, you'll just use one node. There are some cases where you could request multiple nodes at one time. So it's basically submitting 10 SLURM jobs at once. I think that those are more special use cases though. So it's one of those things like if you really need it and want it, you can reach out and we can set that up or show you how to do that. And then the next one, so these are, I'm just going through some of the common environment setup variables, the ones that I use frequently. So I'll go over that, but I'll also show you where you can find more information on these. So here, this -O, this is setting up where, so because we're not running this script interactively, it's not going to just print out updates as the job is running like we would see with a regular bash script. So instead it's going to save that output to this file here. So we need to specify the full path. I'm going to give it my UID. And then you can just give it slurm job dash and then the %J. And so this is a magic variable where it knows to replace the %J with the current SLURM job. And so you'll never overwrite one of these SLURM job output files. And you can alternatively, so I like to just change this to my Scratch directory. But for now, we'll just save these to our home directory in the supercomputer. And then the final basic command that we need to use is this -- partition. So we need to tell it where we want this job to be run. So kind of the main compute cluster is called Notch Peak. So since we're logged in at Notch Peak, we'll be using Notch Peak. But if you wanted to utilize another partition, or if you were on the protected environment to deal with patient data, then you would specify the partition there. But there's also options that you can run that will, like if a job fails or a job completes, you can have it email you and other things like that. So if you just do SLURM commands, this isn't the right one. There is a, here we go. If you go to this website, it will list all the different commands and modifiers that you can use to add some of these other options if they're useful to you. So pretty much any job, any SLURM job that you'll use, any script will have a header like this, exactly like this, or very similar to this. And then something else that we want to have by default is, since we're loading the software that we need through Conda, we'll need to activate the specific Conda environment at the start of each of these SLURM jobs. And so we need to tell it where to find the actual mini Conda execution script. So that is here, but you'll need to, again, switch out this for your UID. So it says this is where to execute Conda from, and then we can do source activate class. And so that will activate our class environment, or whatever you named this Conda environment, you use that. So if you wanted to maybe copy this script that you write and use it for some other purpose, and you want to use a different Conda environment, then you can just modify that environment name there, and then you'll be able to use environment class number 2 or something like that. So this is kind of the standard what you need to have at each SLURM job, and then everything afterwards is similar to your basic bash script, except for you will be using different directories, especially if you're using the Scratch directory to hold intermediate files as you process these jobs. So is this clear to everyone? Does anyone have any questions about kind of the beginning of each SLURM job script? Okay. So so we could build the genome index and process the FASTQ files in the same script, but more often than not, you'll be able to reuse a genome index. And so that's why I like to separate these 2 tasks into 2 separate scripts to kind of partition these jobs. And so that's why we're starting out with building this genome. So this is going to be very similar to that bash script I think that we wrote, or I had you write, to download the source files, except for we're going to be using some different locations since we're dealing with the Scratch directory. And then at the end of downloading those files, we will use star to build the genome index. So we'll just walk through this step by step. So we're going to start with defining those variables that we need. And so I'm going to create this Scratch directory variable, which is going to be that Scratch / general / luster / and then in my case, it's going to be my UID. And we can also, to kind of keep things consistent, we can use the SLURM job ID for if we're processing data. But since we're going to be creating kind of a static directory that will house this genome build, well, I guess I'll just keep Scratch directory as is right there. But we'll add a couple more where we're going to say genome index. We'll call that yeast index. And I like to, so whenever I'm building one of these, I like to include the ensemble build number. So I think right now they're on version 100. And then also, as you'll see later on, depending on the sequencing types, if we did like paired end, 100 base pair, or single end, 50 base pair, that is going to matter for the genome building and aligning. So I like to just specify that. So maybe I'm doing single end 50 base pair, I'll also include that in the genome build, because if I were going to run yeast paired in 100 base pair, I'd have to build a different folder here. And then I will just specify one more. We'll just say reference directory equals, we'll just call this references within the Scratch directory. So now we're going to make sure that these locations are available. And so what I'm going to do is run the make directory -p, and that will just basically, if this file is not available, or if a intermediate file, so maybe I haven't created this references directory yet, it will add all the intermediate files or folders along that path to create this kind of terminal folder here. So I would do something like Scratch directory / ref directory / genome index. So that will, yeah.
What's with the dollar signs?
So the dollar sign is once you've created a bash variable, if you want to use that bash variable, you have to add the dollar sign before it.
Okay, thank you.
Yep. Okay, so then we can just navigate to that directory that we've created. And then I like to, within this file structure, so I'm going to download the GTF file to start with. Let me just grab that URL real quick. So ensemble yeast. Okay. So I would just make sure I'm in this directory, download this file, and then I'm also going to make a directory called genome FASTAs for all those chromosome files. And then I can either just, so I can keep it as is since we're already in this genome index folder, or if you just want to be sure, you can also add the absolute path here just to be safe. But either way should work here. And then we will just navigate to this directory. And then we can run that for loop that we did before where, so either you could have specified the list of chromosomes up here as a variable, or just specify them here. So I'm going to say for X in 1, 2, 3. I'm just going to list all of these real quick. I will do curl OL, and then I'm going to grab that URL real quick. Copy, okay. And then again, we'll just substitute out that chromosome number with that variable, and then done. So this will initialize that genome index folder, get all the reference files that we actually need. And then again, we need to unzip these files. And since I have them in 2 different locations, I am going to run this twice. So I'm going to run this here in the genome fastest file or location. And then I'm going to navigate back a directory so that I'm in just the genome index file for the yeast, and then run this command again so that it unzips the GTF file here. And again, I would add comments throughout here just to kind of organize your thoughts, and just so if you come back to this in a couple months, you know what each step is doing and why you need that. And then the final step that we would need to run is actually building this index with the files that we downloaded. And so, and I'm just gonna, so with this, anytime, oh, that's the wrong. So with any software package, there should always be documentation somewhere. And so if I wanted to know how to build the index, I could open up the manual for star, and then what is this? Genome generate. So if I were to just search this for genome generate, so it'll have a walkthrough of building the genome index, and then what are all the inputs that you would need to be worried about. And so one nice thing too with a lot of these packages is you can run, I guess, sub-jobs in parallel. So say you have a really big task, especially with human, which, I mean, if you're not running this job in parallel, this could take, I don't know, 12, 20 hours on the supercomputer. But what you can do is it allows to, if anyone's familiar about basically how the CPU is set up, you have these threads or cores. And so you can partition sub parts of the job and basically execute each sub aspect of the job on each core in parallel. And so often on the supercomputer, you'll have up to 64 cores that you can be using at once. And so you can basically divide that processing time by 32 or 64 based on the number of cores. Although with a lot of bioinformatics tools and just parallel processing tasks in general, there is this diminishing return. So a lot of software, so maybe the speed boost that you get from running on 30 cores versus 60 cores is negligible. And so it'll kind of limit the amount that you can run to 64. And so if we wanted to, let's see, next shell variable number of cores. So you could specify, so let's just say, we'll say star. With this, it wants to have a argument as run mode, and then we can say that genome generate, so it knows to build the index. And then you could, since based on the core that you run on, or the compute node that you run on, the number of cores may change. I think there's a way to find the exact number of cores, but we'll just leave this run thread number at 32, because I think every compute node in the supercomputer has at least 32 cores. I think after 32 for a lot of these jobs, the return is not as high. And then we will specify where do we want this index to be output, what are the FASTA files, the genome FASTA files, where are those located, where is the GTF file. So this is needed, so this is splice junction, so knowing where the intron spacing is for transcripts so that it can build the index with that in mind, so it's able to align reads that span a splice junction. And then it also wants to know what is that, I guess, what is the average read length. And so generally this is just so we did single end 50 base pair sequencing, and so all it wants is that read length. So it's just wanting to know what's kind of the maximum overhang that I need to be concerned with here. So we will add these options. And so this will just be that path to the genome index file, and then I'm going to put these files in a subfolder, the actual genome index in a subfolder just called genome, and it will create that folder on its own in star. And then we will tell it where are the genome FASTA files. And so those are in this location, the SGDB GTF file. Let me just make sure I'm getting the capitalization correct here. And I guess I haven't, so I didn't specify a variable for the file, but that will just be this bit without the .gz since we've unpackaged that file. And then the SJDB overhang. In my case, since I'm going to be dealing with a data set that's 50 base pair sequencing, I'm going to put 50, but when you're processing your own data, you have to look at the average read size in the FASTQ file to figure that out, or sometimes in the metadata on the geodatabase for that data set, it should include information about that read length. So we can save this and quit. And now we will run the job to actually build this. So to submit a job to the supercomputer, that command is just sbatch, and then the command name, or the SLURM job name. So it's basically like you're running a bash file, except for instead of saying bash, you're going to say sbatch there. So I'll run this. It'll tell you you've submitted that batch job here, and it'll give you an ID. And then you should see as it starts running, you can now see that it created this output file here for that job ID. And so I can look at that file and just see the output of what's going on. So you can see right now it's in the process of downloading all those FASTA files. But also you can check the progress of a job by running SQ. Let's see, -u, so this is going to specify the user. So I would put my name here, and then if I do -w1, so this is going to basically update the progress of this command every second. Okay, maybe not. Let's just run this as is. So it looks like the job actually already failed for whatever reason. So I'm going to open up that progress log again. And so I have an issue where it can't read files from genome FASTA file. So it's saying that it's not finding files in that FASTA file directory that I gave to star. So we can take a look real quick. I'm just going to use one of my shortcuts that I created. So you can see here in the scratch directory, here's that folder that I created, the genome FASTAs file, and all those files are there. So potentially I mistyped the which one was this? I think it was build, and maybe mistyped the location here. Let's see. So scratch directory, ref directory, genome index, genome FASTAs. Interesting. Okay. So I don't know why it's not happy there. So I would have to look back into that. I'll figure out what's going on here with this path and why it doesn't like it. And I will update you all on that. It's very strange. Okay. I don't know what's going on there, but that should be working. Okay. So that's the basics of building a SLURM script and then running that. So say you started running a job and you realized you wanted to modify something, or it was stuck in a loop, but it wouldn't quit out. You can also cancel that job. So it gave us this SLURM ID when it started running, and you can just run scancel with that SLURM ID, and then it'll cancel that job for you. But since it failed, it won't do anything right there. Okay. So now what we can do is create the actual analysis workflow. So taking those FASTQ files, taking the curated genome index, and then processing those files. And so I'm just gonna walk through the steps here since I've kind of demonstrated in real time how that was done. But again, you would create a new file. So this would be process, maybe SRA, and then the ID.sh. And then just like we had done before, we'll add those same SLURM job headers along with the instructions to activate the Conda class or the Conda environment. And so this should include all of the software like STAR and some of the other tools that we'll be referencing in here to be able to actually process the files. And so here I've specified the scratch directory, and here I'm going to use the variable that's created automatically by SLURM that specifies the SLURM job ID. And so in the scratch directory, it will create a directory with just the name of the SLURM job that was executed. And then we can make that directory just like before. And then I'm going to specify as variables the input location, so where I've stored the FASTQ files that I downloaded, in this case from using that SRA toolkit. I'm going to specify the reference directory, so kind of the top level yeast version 100 directory that I just created. I'm going to make a variable for the GTF file. So using that reference variable with the GTF file name, I will have the list of files that I'm going to be using. And so here, I'm not going to specify the .fastq because I'm going to want to reuse just the file IDs, the file names, and be able to swap out the file suffix as I go through the different steps of going from a FASTQ file to a BAM file to a tab separated file. And one other thing I like to do when I'm setting up this scratch directory for the scratch job is specify, kind of organize the input and output files. So within the scratch directory, which is just that in my scratch directory with the job ID, I'll add a file that's called input or folder I'll add one that's called output. I will copy files from my home directory, the FASTQ files. I'll just copy everything from that directory, which should all be the FASTQ files that I want to work with, and copy them to that input folder in the Scratch directory. And then within the output directory in the Scratch directory, I'm going to create subfolders for all the intermediate steps. So once I've pre-processed the FASTQ files, those will go here. When I've aligned those files, I will put them there. When I've sorted the BAM files, I'll put them here. When I've quantified the read counts, I'll put them here. And then any quality control I'll put in this directory. Oh, and then here, so this output variable here, this is a folder that I've created in my home directory, so that at the end of processing the job, I can just copy the final files that I want back to my home directory before I delete the Scratch directory to clear up that Scratch space. And then I'm just going to navigate into the Scratch directory here before I start actually running the processing jobs. Okay, so and then here I'm going to, similarly to how we did a for loop, to process a list of variables. So I want to process that list of sample IDs. So here I'm going to start this for loop. And just to make this more readable, I'm going to put these on different lines with indents so that I can organize kind of what's going on here. So I will do for file in that files variable that lists all those IDs. I'll do semicolon do. And then for each step, I'm going to just echo to the SLURM job output file. That's just going to give all that output that would have gone to the command line. I'm going to say I'm processing this file. I'm running the pre-processing step. So here we'll be using FastP. So say FastP, we'll give it a number of threads to use. I think FastP can only run on 20 threads. We will say that the minimum length that we want to keep is 30 base pairs. So after it's removed the adapters and everything, we'll run that, and then we will trim. So any base that has a Fred score under 28, we will not keep that base, and so that will be removed there. And actually, I forgot to add the here, so I would actually add something like so. FastP thread 20. To say length is 30, quality is 28. And then whenever we want to specify a new line, it's good to have this new line of the same command. We want to add this /, just to say, okay, you're going to continue processing this command from the next line. And then I should add something like -a. And then for that sequencing library prep, whatever the adapter sequence was for that, I'm just going to make one up here. We want to specify that adapter here. And then we'll tell it where to find that input file. So this is in that input directory. We're going to use just that file ID like before, and then add the .fastq there. And then I will specify the output here, which is going to go to that pre-process folder. Again, just use that file ID .fastq, and then it's going to output some kind of quality control metadata for the run to these file types. So you also need to specify this -j and -h. So I'm going to send those to the pre-process folder. Also give it those suffices to output all those files. So I think with a lot of these other steps, you'll be able to find out the meaning of these by looking at the help menus or the manuals for these files. But I just want to quickly go through these other steps for what's going on here. So this is, I should say, fast QC. So this is going to run quality control on the raw reads after removing adapters and removing just bad reads in general. And so with this, you just tell it what's the FASTQ file that you want to be running. So we want to grab the FASTQ files from the pre-process folder, and then we will output these to that QC folder, and we'll just use the file name or the sample ID as the file name there, and then it'll automatically append the suffices that it needs there. So then moving forward, then we would go to aligning. We would again provide that splice junction variable that we provided during index building, and this needs to match what was used when building the index in the first place. So if you were trying to run 50 base pair single-end sequencing on an index that was using 100 base pair splice junctions, they would be incompatible, and you would need to rebuild the index. So then we'll output unmapped SAM files within the same BAM file. We'll output this as a unsorted BAM. We will also, we can do a preliminary quantification of the reads using this quant mode transcriptome SAM. So I'll output another SAM file or a BAM file that is aligned to the transcriptome itself. We'll tell it where to find the genome index, where to find the GTF file, and then we will tell it where to find the input file for this alignment. So that was that FASTQ file that we output to the pre-process folder. And then we will tell it what is the output file prefix. So we need to specify the output location along with that file ID. And then it'll automatically append this _align.bam to each of those files. So then we'll sort that file, specify the output. We'll put that in the post-process file _sorted.bam, and then space, and then the input file, which was that align file from the previous step. And then we'll run the index step. So this will allow us to use that interactive genome visualization tool later. So that just takes the index module command along with that sorted BAM file location. And then we will count those reads that were aligned, basically quantify per gene how many reads align to that gene. And then at the end, I will just run this multi-QC tool on the output directory. So this is going to collect all of the quality control data from this run and compile that. And then I'm going to move all the files that I want to save from their respective location. And then I can just specify I only want files that end with _sorted.bam. I want all of my count files. I want my quality control files. And then I'm just going to output them to that output variable, which I specified up here. This will be on my home directory. And then at the end, I'm just going to remove the scratch directory. I'm going to add this F command right there. So this will force the deletion of the scratch directory. And then that will finish up that script. So that should process all that data. And then again, here are some useful commands for running the job. I guess with this should be not job ID, but that should be the script name. And then scancel job ID. You can see the jobs in the queue. So the position of your job in the total queue of all the jobs waiting to be run on the supercomputer. And then if you want to watch, oh, okay. So that SQ command I ran here, so that is showing all your current jobs. And if you add to the beginning watch-n1, that will update the SQ command every second. So you can get a real-time update on, is the job still running, or did the job quit for some reason? I think in the interest of time, we will save the copying files back and forth between the supercomputer and your computer, and then also visualizing BAM files in the context of the genome for Monday's class. And so the homework for until next time, or for this session, is to basically build those scripts to build the index and process those files for your organism and for the FASTQ files that you downloaded. And then we will save this for next class, where we will transfer the alignment files and be able to actually visualize them on your desktop. So with that, I will take any remaining questions, and I will update some of these commands that weren't working or were mistyped and add that.
Hey, Jordan.
Yeah.
So I have some confusion here. So the genome index, which we are building, how is it different from the reference genome that, I mean to say, is it same as the reference genome that we got last time for a model organism, or is it just now in a script mode or something?
Yes, that's a good question. So it's not the same as those reference files that you initially downloaded from Ensemble. So we can actually go to, so I output this to the genome file. So that's what was generated by STAR. So if we go to the STAR index, oh, I see it didn't build that. Okay, I'm gonna go to one of my old ones that worked. Okay. Oh, wow, I have a bunch of broken genome builds here, apparently. Oh, wow. I wonder if they removed my files since they're old. Okay, no, these are working. So there's going to be a series of files. So basically, the genome index is basically breaking the overall genome down in a way where it can do a faster search through the genome for where your reads should align. And it also has that information of where are those splice junctions for the introns. Yeah, I'm sorry that it looks like my old files got removed, so I can't show you exactly how that's working, but I think if we go, let's see, back to the last class, I think I gave an example of kind of how you can conceptualize this, where, so your initial file is just one long string of nucleotides, whereas the genome index is breaking this up into substrings that it can quickly find out if the read aligns to. If not, go find something else. And it's designed in a way where it can just basically look at, okay, what's the first nucleotide of this read? And okay, now here's the 100 sections in the genome that matches up with that. And then within these, what ones also match the second nucleotide, what matches the third. And so it kind of works in this tree structure where it can quickly search the right location of that nucleotide. Does that make sense, or is that?
So last time we had GDF files like downloaded for the ref genome, right?
Yeah.
And so it's gonna take that into account for building that genome index.
Yeah, so it's in the case of RNA-seq alignment, since there's those reads that may span an intron like here, like we see here.
Yeah.
So that's just telling it basically just creates a resource for STAR that says, if you have a read that is aligning in this region, there's an intron here. So check to see if maybe it can be split between these two exons that are separated by an intron.
Okay.
Does that make sense with that?
I think so.
Yeah, and so it will also in the index, instead of just having like one reference section, so these are all chunks of the genome. So generally without this splice junction, it would just take this and then this and then this. But one of these references will be like this section plus this section, so that that read can align directly to that kind of merged exon.
Okay. Yeah. And then I have actually many more questions,
Okay.
but do you think I should ask in the next session, or like how do you want to like?
You can go ahead and ask right now, since we're not doing that review session today.
Okay.
Maybe we can spend another 5 or 7 minutes on those, and then I'll need to get going.
Okay. And so the thing is, like, what I did yesterday was like I followed all of the last lecture and downloaded, did the prefetch thing and fast them and got all SRR files. So my first question was like, so you said like those SRA files should be downloaded to the NCBI folder, which will automatically be created by the SRA toolkit. So, but what happened with me was like those files, like they were prefetched. I just gave the whole, made the whole bash script for the 8 files. They were downloaded, but like each file made a separate folder on its name in my home directory. There was no NCBI folder generated.
Okay.
And then for the second part, FastDump, what I had to do was like I, instead of giving the output, the input directory address as the NCBI folder, I had to specify it till the end of my home till my home directory, so that it can pick all those files from there. I got all of those files, but I also got some errors, right? And I went to the final output directory and checked for like FASTQ files, and I had all those files there. I made a Word document for that. Maybe I can send you that, and that'll make it more clear.
Yeah, that sounds good. So let me just try this file real quick and see how this is looking. This might take a while. I think one option with this is to specify the output directory with the prefetch. So at the end here, you could say like -o and then you could specify a SRA folder that you want to output everything to.
Okay.
I'm not sure though if that will solve the issue of it making a folder for the file. Like, so I actually have an old file that looks just like that. Yeah, so it downloaded it right there.
Yeah, I checked my home directory for any NCBI folder. I didn't see NCBI folder, but there was a hidden .ncbi folder. And then I CD'd into that, and then I don't see anything like in that.
Okay. Let's try if I just do prefetch -o, and we'll just send it to the home directory. Maybe you have to specify the file name here.
And also, like, once I'm done, can I clear those SRA folders? Because last night I got like a angry mail from CHPC saying that you are over your 122 GB limit, and then they gave me a penalty for like 30 minutes or something. So I think I am almost there. So should I remove those, since we already have those FASTQ file in the final folder?
Yeah, so once you have the FASTQ file, you can delete the SRA file.
Yeah, because they are like, I think 2 GB, 3 GB each or something, and they are taking space. So maybe.
Yeah, that's fine. The only reason you need the SRA file is to generate the FASTQ file afterwards.
Okay, so I can just remove them. And then I uploaded a Word document in the chat box. So maybe that will explain what I was saying, and then also what errors I was getting.
Okay. Is this the one that you sent yesterday afternoon?
No, I just uploaded this FASTQ dump problem.docx in the chat window.
Oh, chat window. Okay.
Yeah.
On the, oh, okay, I see.
Oh, yeah.
Okay, let me open this. Okay, I will read through this and see what I can find. Let's see if this, oh, interesting.
The weird part was I could get all the files, but there was these errors also.
Okay.
I checked head of one of the file, it looked fine to me.
Yeah, so as far as the first question about creating the folder, so if you run prefetch with the -o and just whatever directory you want to put it into, and then the ID with the SRA at the end, that will create it directly in that file, not create the folder, the intermediate folder.
Okay.
And then potentially that will fix the issue of the FASTQ dump. So I can, let's just try this real quick. So I think with the output directory, I'm just going to output that there for now. And then the split file. SRR. Oh, I guess I need to move this. Accidentally named that the wrong thing. So I think running this, at least within this location and this setup, I think that should hypothetically work unless it may be like an issue with, are you downloading these within a bash script?
Yeah, I took 8 or some sample set for 8 or something, and then
Okay.
did a bash script for that.
Do you mind in the Slack channel just sending me directly the, can you just like copy and paste what you wrote for the bash script?
Yeah, I
Okay.
can. I will copy the bash file probably in a Word document or something,
Okay.
or
'Cause then
copy it and or just paste in the Slack channel.
yeah, so it could also just be an issue of how you're referencing the shell variable. And so if I see that, I'll be able to determine whether that's an issue or not.
Okay, what I'll do after the class that I'll copy that and then send it to you in the Slack command line channel or something.
Okay, that sounds great.
Yeah. So yeah, I
Okay.
think these were the major issues. And are we? So we are having 5 p.m. class, or we are not having that?
Yeah, so I'm also holding, so it's basically going to be exactly like this class at 5 o'clock again.
Okay.
So if you're more than welcome to come to the 5 o'clock one as well if you want to listen to all this again.
Okay.
But you don't have to. You don't feel obligated to either.
You know, I think, yeah.
Yeah, it's not going to be
So,
a general review though. It's just going to be these slides again.
Okay. Okay, got it. So yeah, thanks, Jordan. I'll just copy it and paste it in the Slack channel, and then hopefully that
Okay.
may explain better.
Yeah, sounds good. Thanks.
Yeah. Great. Thank you.
Yep.